Hello, everyone. Thanks for coming. My name is Tzu-Kuei Lee, and I'm a graduate student in the Department of Biomechatronics Engineering from National Taiwan University. And this morning, I'm going to share with you about my research. And the topic is three-dimensional shape variation analysis on Sinningia Speciosa flowers. So, in the beginning, I would like to talk about why we did this research. As we know that the studies of biology can be separated into two parts, which are studies of genotype and studies of phenotype. And the shape analysis of organisms plays an important role in the studies of phenotype. But most of the previous research focused on two-dimensional shape analysis instead of three-dimensional shape analysis. So we think quantifying three-dimensional flower shape might be a challenge and also interesting topic for study. So we choose Sinningia Speciosa as the samples of our research. Sinningia Speciosa, also called Darwin Glossinia, is a kind of flower. And this species has two types of flower with distinct floral shape. And one is the wild type form, and the other one is caloric form. And as this picture, you can see the most significant difference of shape between them is the floral symmetry. The wild type form belongs to the bilateral symmetry, and the caloric form belongs to radial symmetry. So from previous research, we know that there is a phenotype called podia, which affects floral shape and symmetry. So the wild type form, this in bilateral symmetry, corresponds to the site allele homozygous to wild type. And the caloric form in radial symmetry corresponds to the site allele homozygous to caloric. So here we consider these two types of flowers as parent and intercross them to breed the F1 flowers, which corresponds to the heterozygote genotype. And by selfing of a single F1 flower, we will get the F2 flowers and in three different site genotypes. And as this picture, you can see the floral shape of F2 flowers are quite varied due to the interbreeding process. So this is called the floral shape polymorphism, which is able to provide an opportunity for us to analyze their floral shape variation. But here comes the challenge. For these numerous and variously shaped F2 flowers, how to quantify 3D floral shape and analyze floral shape variation. So our objective is to quantify floral shape variation among F2 flowers of Sinningia Speciosa and to analyze the variation of the floral landmarks using geometric morphometrics. And also, we would like to find out the association between floral shape variation and site genotype. So this is the procedure of our research. Firstly, we will acquire our 3D floral images, and next identify the floral landmarks, which are applied to describe the floral shape. And by doing geometric morphometrics, we will analyze the floral shape variation in the last part. So this slide shows how we get our 3D floral images. 39 samples randomly selected from F2 flowers were used for scanning. And we choose micro-CT as the scanning equipment. The advantage of micro-CT includes high scan resolution. We use 35 micrometer scan resolution, which is high enough for shape analysis, and non-destructive. And also, the images from micro-CT can accurately present original objects instead of distortion effect. So the right picture shows the 3D volumetric images from micro-CT. So once we acquire the 3D floral images, we will use landmarks to define the floral shape of individuals. And there are two types of landmarks. The first one is primary landmarks, which is defined as homologous points on biological structure. And the other one is secondary landmarks, which is defined as points uniformly distributed along the contour between two adjacent primary landmarks. So this slide shows an example of how we identify the 3D floral landmarks. First, we identify the primary landmark, which is the black dots with numbers shown on this picture. And then we identify the contour, which corresponds to the low contour and pattern grid shown on this picture. And then the secondary landmark, which corresponds to the white dots, here shown on the picture, could be identified. And note that the color area only shows the floral landmarks of single petal. So according to this rule, we could further identify the floral landmarks of complete flower. So this slide shows the procedure of landmark selection. First, it is the 3D volumetric image from micro-CT. And next, we use a method function called isosurface to extract the surface image. And then we use a software called landmark to identify the contours we need. And finally, we will get totally 95 floral landmarks for each sample. So after landmark selection, we will go to geometric morphometrics, which is a method for analyzing shape variation. So basically, geometric morphometrics include two steps. The first step is general precocity analysis, also called GPA. The purpose of GPA is to eliminate variance irrelevant to shape, which include the effect of translation, rotation, and scaling. So this is an example of GPA. These are two initial configurations. And after translation, rotation, and scaling, the irrelevant effect will no longer affect the final shapes, which can be compared more effectively. So the second step of geometric morphometrics is principal component analysis, also called PCA. The purpose of PCA is to find the most significant shape variation among F2 flowers. And each PC would correspond to one independent shape variation. And the first few PCs will represent the most of the variance of floral shape. So this table shows the variance percentage of PCs. And we can see the first two PCs, which account for 41% and 20%, are much higher than other PCs. So we can say the first two PCs could represent the major shape change among F2 flowers. So here I'm going to introduce how to visualize the shape variation in PCs. So this is a scatter plot of PC1 and PC2. Firstly, we identify the mean shape at the zero position of each PCs, which is over here. And now if we want to know the shape variation in PC1, we would have to identify another two PC values, which refer to -2 standard deviation and +2 standard deviation. And then reversing the PC values to get the virtual flower like this. So by doing the process of reversing PC values, we would be able to easily recognize the shape variation in PCs. So this slide shows the visualization of floral shape variation in the first two PCs. As we just mentioned, the first two PCs can represent the major shape change among F2 flowers. So for PC1, the shape change corresponds to the level of petal recurving. The flower with small PC1 values have lower degree of petal recurving, while the flowers with large PC1 values have a higher degree of petal recurving. And for PC2, the shape change corresponds to the level of closely-ventral symmetry. The flowers with small PC2 value belongs to the radial symmetry, while the flowers with large PC2 values belong to bilateral symmetry. So in the last part of our research, we would like to know the association between site genotype and PCs. So this is a PC1, PC2 scatter plot with PC histograms. And note that the three different site genotypes are in different colors shown in this figure. So here we use ANOVA to determine the association between the shape variation and the PCs. And the null hypothesis of ANOVA is defined as PC values in different site genotype are set. And the confidence interval is set to 95%. So the result of PC1, you can see the p-value is larger than 0.05. So the PC1 is not significantly associated with site genotype. And for the result of PC2, the p-value is extremely small. So it indicates that PC2 is significantly associated with site genotype. So here comes our conclusion. The floral shape variation in F2 flowers of Sinningia Speciosa is quantitatively evaluated. The shape change of PCs can be visualized by reversing PC values. And the quantitative evaluation of shape variation, which is the approach we developed, can be applied to the floral genotype-phenotype association. So this is the end of my presentation. Thank you for listening.
You didn't use other 3D cameras other sensors to measure the shape. Instead, you used micro CT. Micro CT, I assume it's more expensive. What's the cost of micro CT?
The cost is actually is not so much. Maybe $30 US per hour.
Okay.
Yeah.
So yeah, have you considered using other 3D cameras?
We think because we want to acquire the very accurate 3D images, and we think that 3D camera can reach our goal. Can reach our goal. Because it's kind of some, I think, distortion, not so accurate. And micro CTs can present a very accurate image from original objects.
What's the speed of your scanning micro CT?
It's a little bit time consuming. For one floor, like 30 minutes to 1 hour.
Other questions? Are you going to be adding any color information? Right now, everything is based on shape at this point?
Yes.
What about color?
We have planned to quantify the color.
Mm-hmm.
Yeah. Maybe in the future.